Hi there and welcome to this IB revision video for IB Biology, and this is a HL only video looking at the origins of cells and water on Earth. So let's have a look. First of all, then you need to know the conditions of the early Earth and the early atmosphere. And obviously life was a lot more tricky in the early atmosphere. We think life arrived probably on an asteroid. Potentially due to the primordial soup in the Earth, which we'll talk about as we go forward. What was this primordial soup? Where did these chemicals come from that could create organic matter? Well, the atmosphere basically would have originated from volcanoes. The Earth was much more radioactive, much more volatile at the time, and therefore these main chemicals were being released: so methane, water, carbon dioxide, nitrogen and ammonia. And this was due to intense volcanic activity, and it led to a similar atmosphere as what is on Mars and Venus today. There was no ozone, so no O3 at the top of the atmosphere protecting us from radiation, and therefore all this energy was present as well, because UV radiation was on the Earth. The biggest change that happened, first of all, I suppose before this one, the Earth cooled down. So the Earth must have been above 100 degrees because water vapor was all vapor, it was all gas to start with, all steam. The Earth then cooled down, forming the oceans. Once the Earth formed the oceans, somehow life evolved on planet Earth. And it did this through algae and probably photosynthetic bacteria, first of all, doing photosynthesis, and then very early aquatic plants started doing photosynthesis before life evolved onto the land. The water would have protected those plants from the ozone. You need to know the equation for photosynthesis, hopefully you already do anyway. And algae, by doing this, would have started to reduce the carbon dioxide in the atmosphere, which allowed the temperature to drop further and increase the amount of oxygen in the atmosphere. And over the next billion years, land plants started to evolve as well, increasing the oxygen gradually in the atmosphere. A lot of this oxygen at first wouldn't have just stayed as O2 in the air. It would have reacted with each other, especially in the UV rays, to make O3, which is ozone, which would then protect the planet against UV radiation. So we need to know then about the cell, if we're going to look at the origins of cells. And the cell is the basic living structure of all living things. It can't get any smaller than a cell than be alive. Cells are living because they use energy to maintain an ordered state. If they stop, they'll be dead. And cells can divide to make more cells, and we know they can be taken from the body and be kept alive if kept in the right conditions. Whereas a virus is not a cell, it's acellular and non-living because it doesn't have a plasma membrane, it doesn't have any cytoplasm, and it doesn't often have DNA. Obviously some viruses can have DNA, but it might not, it might have just RNA. It is just literally a protein capsid surrounding some genetic material. It has no independent means of metabolism or growth, and it needs a host to reproduce. So where do cells come from then? Well, this is obviously, we always say that in the cell theory, a cell must have developed from a previous cell. So I will be now something like 1 billion cells, and I originated from lots of previous cells, and they originated from the zygote, which came from a sperm and egg cell, etc., etc. And that came from my parents, which then came from my grandparents. But at some point, the very first cells must have developed. So Louis Pasteur said that life cannot be spontaneously produced, and he did an experiment looking at decay and microbial growth. And what he did was he used this piece of equipment here, where he used this, he boiled all the material in here, and it had this sort of U-bend here stopping any new bacteria getting in, and he waited for a long time and he found no growth was there. If he did the same, killing all the original life in here, but broke the neck of this jar, he would later find that microbial growth would develop. And therefore it shows you that if the microbes can get in, they'll grow, but if there's nothing in there in the first place, nothing will actually develop. But that leads us to the point of the cell theory, which all cells arise from pre-existing cells. As I said a few moments ago, where did the first cells come from then? They must have developed from some form of non-living material. So what needs to be present for cells to be alive or to develop or to evolve? Well, there needs to be some sort of control of reactions, so some sort of catalysis. There needs to be some sort of self-assembly. The monomers need to form into polymers. There needs to be compartmentalization, which means the membranes are needed to separate contents from the outside world to the inside world to different parts of the cells, and there needs to be some sort of inheritance mechanisms of replication of molecules. So the basic question is, well, how do these simple sugars that may have evolved turn into complex complex molecules, which could then become a primitive cell? So where do we have any evidence that this could have happened in some sort of primordial soup? Well, the evidence for the origin of these, again, they're all carbon-based compounds, has been looked into. So in 1929, Haldane came up with the hot dilute soup theory, and he said that before life there was a prebiotic, so pre-living ocean full of carbon compounds from the atmosphere, full of water, vapor, carbon dioxide and ammonia. And if you think about it, those are the chemicals that make life, because carbon dioxide has obviously got carbon and oxygen in them. Water vapor has H2O, so there's your hydrogen source, and ammonia is NH3, so that's got nitrogen. So these are the main four chemicals in 99% of all living material or all organic molecules. He claimed that if you were to shine UV light on them, they would eventually form sugars and proteins. But do we have any evidence that this could happen? Yes, we do. So something happened. Miller-Urey did an experiment later on, and they did this experiment here. So what they did is they set up this experiment to represent the conditions of the early life on Earth. So here we have the initial compounds that were added into a big glass bowl, and that was to represent the gases in the early atmosphere, which I discussed a few minutes ago. He then sparked lightning through these because it was electricity to represent lightning. Okay, and then he passed these gases through a condenser to turn them into a liquid, and then basically cooled them down to try and sample and see if anything was collected. He had water in steam to give a constant supply of water and circulation of that water to represent the early ocean and the early temperatures of the Earth. And basically what Miller-Urey found was that if left over time, this created a variety of carbon compounds and, more importantly, all 20 amino acids. So what this shows us is that the complex organic molecules can be created from simple inorganic molecules without life, and this could then have triggered the first forms of life to occur, the first cells to occur, maybe even probably simply by accident. Now, you need to make a cell, you need a sort of membrane around all these different chemicals. So how were these vesicles forming and how did molecules form? Well, the idea is vesicles are small droplets, and because phospholipids are amphipathic, which means they have a hydrophilic and a hydrophobic part, they will react to water. So what this can lead to is the formation of micelles. And even though these are not proper cells technically, because they're not double-bounded membranes, they are enough for maybe the cells to have developed. And maybe in these areas here, some simple compounds of versions of proteins and amino acids got trapped, and then that became the first forms of life. And obviously over time, the membranes has developed. So that might be then how the first membranes developed. In terms of genetic material, it is believed that RNA was probably the first genetic material. And that hopefully makes sense because RNA is simpler, it's quicker, it's short-chained, and therefore it's easier to replicate. It has a high mutation rate, okay, and that would have aided simple variation in early life. And as life became more complex, this mutation may become a disadvantage, and therefore it became more advantageous to have DNA which is more stable. RNA can also act as a catalyst in some reactions. So it might be that the RNA was there to act as a catalyst, and it can be used to form peptide bonds, for example, between amino acids. Now, to understand this a bit more and think, well, how does this all work? We need to look at this genetic code and use it to look at something called the last common ancestor. So we know that the genetic code is universal. We know it codes, so it's a degenerate code. It codes with 20 amino acids with 64 different combinations. And the fact that all living things use this code today shows that there must have been a last common ancestor that we all had. And looking at this a bit more detail, if you look at the structures of key things, we have the same genetic code for key structures. So for example, the enzyme cytochrome C used in all respiration, that has the same DNA code. And 350 plus proteins can be traced back to the last common ancestor, called the last universal common ancestor, or LUCA. Other organisms with different genetic codes were believed to be outcompeted by the more successful LUCA. So what this means is that we know at some point all of our DNA has come from a common ancestor, which would be millions and millions of years ago. And it might be that before that there was other alternative versions of life that died out or outcompeted. So we can use this to work out that we must have had a last universal common ancestor a long way back, hence all the variety today has the same genetic code. And one area where we think this may well be is the Strelley Pool stromatolites in Hamelin Bay, Australia. And this is the earliest evidence of life on Earth. And what these are is cyanobacteria. So these, what look like rocks, are actually calcium carbonate deposits released by cyanobacteria over thousands and thousands of years. We can radioactively date these to identify that they are about 3.42 billion years old, but we think even simpler cells must be a lot older than this. But that's our evidence for the oldest life we can find. The oldest rocks we have do not contain fossils, but they have a carbon ratio for isotopes that is low and suggests that life is present, and life with the right ratio have been found up to 3.85 billion years ago. Some suggestions that rocks in Australia with depleted carbon may be 4.1 billion years ago, but there is limited evidence of that. So we think cells evolved over about 4 billion years ago, and that is as far back really as we can go. Now, there are also hydrothermal vents, and hydrothermal vents we feel are in deep ocean, so extremely sort of cold anywhere else, but really hot by the hydrothermal vents, high levels of chemicals that would be toxic to us, and low levels of pH because we think they're actually alkaline, high pH because we think they're alkaline, had similar conditions to the early conditions on Earth where life evolved. And now scientists feel that this may actually be where life first evolved on the planet, because they gave the similar conditions to the Miller-Urey experiment I described earlier. Now, the other thing then you might be talking about is, well, where did the eukaryotic cells come from? Now, eukaryotic cells, again, are cells with nucleus and cells with membrane-bound organelles of a larger size. So they are ATS and ribosomes, they've got larger ribosomes, but also they could be plants, animals, fungus, or protoctists. And we think they evolved through something called endosymbiosis. So endosymbiosis is when one organism, the endosymbiont, lives inside another organism, a host. And basically it will be then using some of the material. So it goes in via endocytosis through a vesicle. So if you have a look at this example here, this is what happens in phagocytosis, and the pathogen might go in via a vesicle, and then the phagocyte digests it. But in this case, it doesn't digest it. It might be that the organism remains in the host. If the host benefits from the ingested organism, it will not digest it. So it's a mutualistic relationship where both organisms benefit from it. And what we think probably happened was that basic, simple cyanobacteria, bacteria that can do photosynthesis, would have been endosymbiosis into original larger cells, and that would have formed, and they would have then become chloroplasts. So they would have provided the cell with glucose from photosynthesis, and the cell would have provided them with the materials they need to survive as well. And again, aerobic bacteria that could do respiration might have also gone through endosymbiosis into the cytoplasm of pre-existing larger cells, and that again might have allowed them to survive. They provided the cell with ATP, the cell provided the bacteria with what it needed to survive. So it's a mutualistic endosymbiotic relationship. Now, over time then, that's how we think eukaryotic cells have adapted. And one of the pieces of evidence that this could be the case is that chloroplasts and mitochondria have their own DNA, but it is very similar to prokaryotic DNA. So it's naked DNA, circular DNA, a lot more smaller DNA chains. So we think, as I said a moment ago, mitochondria are free living prokaryotes that could do aerobic respiration. Larger prokaryotes then could only do anaerobic, and they link together to get that more energy for the larger prokaryotes. The larger prokaryotes would have provided it with more protection and supplied it with the materials it needed to be alive itself. Smaller cyanobacteria then could probably do photosynthesis and have the same process occur as a result. So what is the evidence for this? Well, this is under the following areas. So there is components, different components we can look at to provide us with evidence. So membranes. So the first piece of evidence is membranes, and the fact that membranes in eukaryotic cells are double membrane bounded would suggest that they were originally, let's say you had originally a mitochondria that come in. Because it was inside a vesicle, it has become double bounded as a result. Antibiotics, some organelles have parts that are susceptible to antibiotics, suggesting bacterial origins. Division, reproduction occurs of these part of cells in a fission-like process similar to prokaryotic cell division. DNA has each cell, these areas of these cells, organelles like I said before, chloroplasts or mitochondria have their own DNA, very similar to simplistic prokaryotic cell DNA. And ribosomes, the chloroplasts and the mitochondria have ribosomes which are 70S in size, so it's similar, identical even to prokaryotic cells, which they use to make their own proteins to control photosynthesis and respiration. So they're almost self-sufficient, but not quite anymore. There you go. So that is the information on endosymbiosis.